Hi, I'm David Rimm. I'm professor of pathology at Yale University School of Medicine. I think the future of the industry for antibody production is represented in three parts, and the part that I'm most familiar with is the diagnostic antibody or the scientific antibody aspect of the industry, and I think that aspect of the industry will change over the years as we become more standardized, as we work toward more recombinant antibodies and away from the spray of antibodies with very little validation and, in some sense, trying to make up for in quantity what we lack in quality. The most interesting work that's probably being done now is looking at ways of producing antibodies that have defined epitopes and defined stability, so that in the therapeutic realm this has been common for a long time, but now moving into the diagnostic realm, trying to produce antibodies that can be part of FDA tests, FDA-approved tests that ultimately can define with some specificity and sensitivity which patients will respond to a given therapy. Biggest growth opportunities are probably in making antibodies more reproducible and more standardized, and I think a lot of companies are realizing the importance of generating the antibodies in ways that increase reproducibility. So I think we'll see more companies that are moving away from animal-produced antibodies and moving toward recombinantly produced, completely sequence-defined reagents. Well, within the next year or so, actually next September, I believe, there will be a stakeholders meeting for people that use antibodies that will be held at Asilomar in the vein of the Asilomar recombinant DNA conference from years back, with the idea that we'll be able to figure out why we've had historically these antibody reproducibility problems and some sort of come up with some series of standards or best practices that will hopefully be widely appreciated and in some sense required by funding organizations and organizations that publish results, so that we can come up with data that uses antibodies that's more trustworthy and less likely to be fraught with error compared to what our historical usage of antibodies largely was.